Alrighty, we're going to start chapter 6, which is the muscular system. Let's start with a little breakdown of muscle tissue. Muscle tissue in general allows for body movement as well as organ movement within the organs. Altogether, we're talking 40 to 50% of your body mass is made of muscle tissue. It has a very limited capacity for regeneration. Only certain types of muscular tissue can regenerate. Mostly just the smooth muscle tissue can regenerate. Skeletal muscle and cardiac muscle have a more difficult time regenerating. Cardiac muscle will not regenerate at all. The three types of muscle tissue, if you recall from our chapter 3 when we talked about tissues: skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle is found around the skeletal system. Here it's responsible for our body movements, which include voluntary and involuntary body movements. Voluntary body movements are consciously controlled. You consciously have to think about moving your arm to scratch your nose. Involuntary movements are unconsciously controlled. You don't have to think about your heart contracting every time it beats, and you don't have to think about your smooth muscle and your digestive system contracting to propel food through. The skeletal muscle also helps with skeletal stabilization. Hold on, I think I just confused you with the discussion of involuntary skeletal muscle. I just realized that involuntary skeletal muscle would be like a reflex, where your body's responding automatically. Sorry for that misexample there. The skeletal muscle also is responsible for skeletal stabilization. Think about your core, think about the muscles of your back holding everything up and in place, as well as heat generation. With muscle contractions, you get increase in friction. Increase in friction is going to create heat for the body. The skeletal muscle is also striated, has a striped appearance. You can see that when you look at it under a microscope. And as I mentioned, the regeneration is very limited after an injury, although it can regenerate slightly. Cardiac muscle is the type of muscle that's found within the walls of the heart. It's responsible for generating the force needed to pump blood throughout the body. This is under involuntary control, so the ability to contract is actually done without input from the nervous system. It has its own conduction system within it, and we'll look at that when we get to the cardiovascular chapter. Cardiac muscle is also striated, so it has that striped appearance under the microscope, and it can regenerate after an injury, but it's very, very minimal, if any, which is why individuals that suffer from heart attacks have a tough time regrowing their heart muscle tissue. Smooth muscle is a type of muscle that forms around the hollow organs such as blood vessels, intestines, and uterus. It's responsible for moving substances through the body as well as regulating the overall volume or size of the organ. It also helps to store substances. It's involuntary. You don't have to think about moving these smooth muscles. They're non-striated and they're smaller than other muscle cells. So they don't have that same striped appearance, and they're small, more spindle shaped. They regenerate more easily than other muscle tissue types because they're not as organized as the other ones. There you can see muscle tissue. This is skeletal muscle, for example, multinucleate, striated, very distinguishable appearance under the microscope. Here you can see the cardiac muscle tissue. It's branched. We also have those intercalated discs, which are special cell junctions in between the cardiocytes, connecting them together so they all contract at the same time. Smooth muscle tissue is less organized. It's more spindle shaped. Only one nucleus within these, and there's no striations or no stripes in their appearance. So let's talk about the overall structure of skeletal muscle. The majority of this chapter is going to focus on skeletal muscle structure as opposed to the other types of muscle tissue. Skeletal muscle fibers are the technical term for muscle cells. Those fibers are bundled together in groups called fascicles. Those muscle fibers or muscle cells, as we've mentioned, are multinucleated. They have a plasma membrane which invaginates or kind of dips into parts of the cell to form a structure called a transverse tubule or T-tubule. Endoplasmic reticulum in this type of cell is called sarcoplasmic reticulum, and it's called sarcoplasmic reticulum because it's slightly different than endoplasmic reticulum of, say, a skin cell. And this is because it holds calcium ions. These sarcoplasmic reticulum are located near those invaginations called T-tubules. and they help or they are stimulated with an action potential from the nervous system to release calcium. Myofilaments are small thread-like proteins inside of the muscle cells or muscle fibers. They will allow the cell to contract and shorten with stimulation. Here you can see what I was talking about inside of the individual muscle fibers or muscle cells. There's lots of myofilaments in there. Those myofilaments are made of myofibrils, and those are just small protein structures that are going to allow the cell to contract. Here we bundle up all of our muscle cells into a fascicle, which is a bundle of muscle cells, as I've mentioned. That fascicle is surrounded by a structure called a paramecium, and then all of those fascicles are bundled together and held together by a structure called the epimecium. It's just another connective tissue layer. The epimecium then forms the tendon, and the tendon connects that muscle to a bone. Here you can see the inside structure of our muscle fibers or muscle cells. Here you see the specialized cell membrane of our muscle cells. It's called a sarcolemma as opposed to a plasma membrane, and it invaginates and folds in, and that's why you see all these blue lines coming in. Those are those T tubules or transverse tubules that we mentioned. Those T tubules butt up against these orange structures, which look like endoplasmic reticulum. That's exactly what they are. Remember, in a muscle cell, they're called sarcoplasmic reticulum, and they store calcium. Those T tubules and sarcoplasmic reticulum line up together and wrap around these special contractile units called myofibrils. And these myofibrils are going to slide across one another to shorten microscopically to form those large movements that we get with muscle contractions. Here you can see the structure of the myofibril. It's arranged of a special organized structure called a sarcomere. This entire structure from Z disc to Z disc is called a sarcomere. This is the contractile unit of the muscle cell. So if we look closer at our sarcomere, you can see that it's made of thick filaments, these dark red lines, as well as thin filaments here, these thin lines. The thin filaments are connected to the Z discs on either side of the sarcomere. The thick filaments are attached to something called an M line in the center or middle of the sarcomere. As our sarcomere contracts, these thin filaments are going to slide toward that M line from either direction. So with a muscle contraction, the Z discs are actually going to squeeze together. You can see down here the more microscopic image of what this sarcomere is. You see these thick filaments actually have little heads on them, almost like little heads of a golf club, and they reach over and grab those thin filaments that are attached to the Z disc, and that Z disc is then going to be pulled toward that middle from both sides. So you can imagine those two Z discs squeezing together, pinching together as that muscle cell contracts. If we talk more in depth about these thick and thin filaments, the thick filament is called myosin. It's a protein molecule. It is a filamentous protein, and on the ends it swells into a structure called a head. So we have myosin head and myosin tail. Those myosin tails bundle together to make the thick filament. The thin filament is made of a few different protein molecules, the main one being actin. Actin has other proteins on it called troponin and tropomyosin. Tropomyosin is part of this filamentous part here. Troponin are these little blue structures. So troponin and tropomyosin are bound together, and then themselves are bound to the actin. The actin would be these yellow circles. So let's talk more in depth about the structure of the sarcomere. As I mentioned, the sarcomere is the contractile unit of a muscle fiber. Stack the sarcomeres end to end to end to end to end, and then bundle a bunch of these sarcomeres together, and that's basically what fills every single muscle fiber. As I mentioned, the sarcomeres are made of thick and thin filaments. The thin filament is an actin protein. The thick filament is a myosin protein. and the Z discs are the boundaries between the sarcomeres. The thin filaments connect to Z discs on either side. Here you can see what I was alluding to before. You see the Z disc and another Z disc, and here's another Z disc. So this would be one sarcomere here, and this would be another sarcomere here. So what we're looking at is two sarcomeres, sarcomere here and a sarcomere here. You see the thin filaments attached to the Z disc. In the center of the sarcomere is the thick filaments. There's our myosin. You can see those myosin heads that try to reach up or down and grab that actin, and then they're going to pull. This is in a relaxed muscle. You can see what it would look like in an actual microscopic image in this picture. If we were to partially contract that muscle, look at what's happening. These myosin heads are grabbing on to that sarcomere, or to those thin filaments I should say, and the distance between Z disc and Z disc is shortening. The distance between Z disc and Z disc is shortening in a partially contracted muscle. Here we have a maximally contracted muscle, and look at how close together those Z discs are now. Our myosin has pulled the actin so close together that they're actually overlapping here in the middle. See that overlap in the middle? That's because those myosin heads reach up and grab those thin filaments, grab those actin filaments, and pull those Z discs into the middle of the sarcomere. This animation here goes through all of the same things that I was just lecturing on, except it's an animation form, so it will review muscle cell structures for you. There's a theory related to muscle contraction. It's called the sliding filament theory, and this is basically exactly what we just reviewed, the idea that those myosin heads, those thick filaments, attach to special binding sites on the thin filaments. Something called a cross bridge forms when the two thick and thin filaments interact. The actin filaments are then pulled inwards in a repeating process called the contraction cycle. Calcium ions and ATP are both necessary for muscle contraction, and those contraction cycles continue until those supplies are depleted in the sarcoplasm, which is the special term for the cytoplasm in the muscle cell. So your muscle cells need two things to contract, ATP as well as calcium. Calcium stored in that sarcoplasmic reticulum, as I mentioned before. That sarcoplasmic reticulum releases calcium. Calcium's then going to allow the actin and myosin to bind together, which then causes the sliding filament theory, that formation of cross bridges, and the movement of those two Z lines towards one another. Here you see the entire chain of reaction that I was just going through. Calcium binds to the thin filaments, revealing the active site. Here we can see our myosin heads attaching on. You can see our ATP, that's our ADP plus P, kind of waiting to be formed there. The myosin heads react, forming those cross bridges. Notice we've used up the ATP. In this case, we're just down to ADP. Now we have our power stroke, or our contraction, where the two Z discs are going to move toward each other. Then those myosin heads are going to attach to a new ATP. That formation between the myosin heads and the new ATP molecule deactivates the cross bridge and allows the myosin to unattach and relax the muscle. This goes through the contraction and movement part of skeletal muscle. So let's talk about the initiation of skeletal muscle contraction. Ultimately, just with all body processes, it's controlled by the nervous system. Nerve signals initiate both voluntary and involuntary skeletal muscle contractions. Where all the magic happens is the neuromuscular junction. This is the interchange between the nervous system and the skeletal muscle system. This is where the electrical impulse from the nerve signal is then converted into a chemical signal that tells our muscle cell to contract. That telling of the muscle cell to contract is called depolarization. Acetylcholine is the chemical signal that is sent from the nervous system cell, from the neuron, to the skeletal muscle. Here you can see the anatomy of the neuromuscular junction. So This is a macroscopic image here of a nerve cell, a neuron coming down. It branches off into several different pieces called axon terminals. These axon terminals swell at the ends, called a synaptic bulb. Those synaptic bulbs kind of hover right over top of the cell membrane of our muscle cell. And here you can see there's a small gap in between the neuron and the muscle cell. Inside these little blue circles on our synaptic bulb contain acetylcholine, ACH. ACH is the chemical messenger that's sent from the neuron to our muscle cell. So you can see here these little purple dots. Those purple dots are the acetylcholine. They're binding to special receptors. You can see them here better. Acetylcholine is going to bind to special receptors on our muscle cell. It goes across this gap called a synapse or synaptic cleft. That's the space between the nerve cell and our muscle cell. And when that acetylcholine binds to those special receptors, it causes our muscle cell to depolarize. The beginning of the depolarization is the influx of sodium ions. So when the acetylcholine binds to the receptor, as we see here, that channel protein is now going to open and sodium rushes in to our muscle cell. This causes an action potential or chemical signal inside our muscle cell. It's called a depolarization. This depolarization now begins that contraction cycle that is needed for muscle contraction to occur. This animation goes through the anatomy and activities of the neuromuscular junction. So, as I mentioned, that electrical state of the cell membrane being reversed is called a depolarization. This is triggered when that acetylcholine binds to the receptor and sodium rushes in. This is going to initiate a contraction through depolarization called an action potential. The action potential travels through that cell membrane into the T tubules, which remember are the invaginations of that cell membrane. If you recall, those T tubules are right next to sarcoplasmic reticulum, which stores calcium. Once the T tubules receive the action potential, that tells the sarcoplasmic reticulum to release calcium. Calcium being released from the sarcoplasmic reticulum can then bind to the troponin tropomyosin complex on the actin molecule, reveal the active site so that myosin can bind to the actin molecule, a cross bridge can form, and contraction can occur. When calcium levels in the cytoplasm return to normal, the muscle cell relaxes, and that is called repolarization. The cell goes back to normal. This is the entire process called excitation-contraction coupling. You see our motor neuron here, the synaptic bulb and the synaptic cleft releasing the acetylcholine molecules binding to the receptors. Once it binds to the receptors, sodium's going to rush in. As that sodium rushes in, it's going to trigger the action potential. That's what these little red arrows are showing. That action potential travels down the plasma membrane or sarcolemma of our muscle cell. It travels into the T tubules, which are those invaginations. Once triggering the T tubules, it's then going to communicate with our sarcoplasmic reticulum to release calcium, these little purple circles. Once calcium is released, it can bind to the active sites on our actin molecule. That changes the troponin tropomyosin complex, allowing for our myosin to bind to the actin. We get cross bridges forming and the power stroke happening. Our calcium is then pumped back into our sarcoplasmic reticulum. The troponin tropomyosin complex resumes its position on the actin molecule. Our muscle relaxes, and the whole contraction cycle can start again. This animation here goes through the complex contraction cycle of a sarcomere. So let's talk about how muscles get their energy for contraction. Ultimately, all cellular energy comes from ATP. Remember that really bad whiteboard drawing that I did of ATP being converted to ADP plus P? Well, the same thing happens every time the muscle cells contract. Another molecule can be used to store energy in muscle cells, and that is called creatine phosphate. Creatine phosphate works the same way as an ADP molecule does. Creatine phosphate, as you see here in the middle, can donate its phosphate to ADP to form ATP. So if the cell is running low on ATP but has a lot of ADP around, creatine phosphate can give its phosphate to the ADP, forming ATP and creatine. This is why if you've ever known somebody who's been into weightlifting or wants to get big, so to speak, you may see them drinking protein shakes that contain creatine in them. The creatine phosphate is going to give their muscles a little extra energy so that they can withstand extra reps at the gym. This is all done through anaerobic respiration and glycolysis, which does not require any oxygen to produce energy. Aerobic respiration does require oxygen for the chemical reaction to take place. So in this case, we're looking at ATP from anaerobic respiration. Glycolysis is the process of anaerobic respiration. In glycolysis, the muscles can break down glycogen, which is where glycogen is stored, if you recall, in the skeletal muscles as well as within the liver. So when your blood glucose levels are depleted, or if your blood is diffusing glucose into the muscle cells, either way, glucose will be present in the skeletal muscle. A process not requiring oxygen can take place as soon as that glucose enters the cell. It's called glycolysis. Glycolysis is going to yield 2 ATP molecules, which can then be used for energy. Glycolysis is going to create 2 pyruvic acid molecules, which, if there's no oxygen present, will be converted into lactic acid molecules, which can be converted into the blood. This whole process takes a little bit longer than creatine phosphate, which was only about 15 seconds. This takes somewhere between 30 seconds and a minute to create. If our body is to use aerobic respiration, you can see here the time limit 1 to 2 hours, maybe even more. This is because it's a highly complex process. We'll look at the details a little later. In short, amino acids from protein breakdown can go into the mitochondria to be used to make ATP, carbon dioxide, and water. Fatty acids can be freed from adipose cells to make ATP, carbon dioxide, and water. Pyruvic acid from glycolysis can be used to make ATP, carbon dioxide, and water. But all of this requires oxygen from hemoglobin or from myoglobin. Hemoglobin, of course, is in the blood. Myoglobin is stored within the muscle fibers. It's what makes our muscle fibers that nice pinky or dark red color. Whoops. As I mentioned, all of these processes require oxygen, which is why they are called aerobic processes. They will ultimately yield more ATP than the anaerobic processes, but again, they take longer. Muscles can no longer contract after a certain period of time, and this is usually when ATP supplies are dipping or declining. This is because maybe a depletion of creatine phosphate, a depletion of glycogen or blood glucose, inadequate delivery of oxygen, maybe your heart just can't pump any faster to get the oxygen through, and possibly because there's been so much lactic acid built up that your muscles are too fatigued to go on. This can cause muscle cramping. When ATP levels decrease and lactic acid levels increase, our muscle cells just can't relax. That's what causes that cramping or Charlie horse sensation. Active transport is then going to work to remove calcium from the cells so that they can no longer contract, but that's also going to require the use of ATP because it is an active transport process. Oxygen debt occurs after lots of exercise as well. Not enough oxygen for functioning due to strenuous exercise and physical activity. If your muscles are constantly acting at peak and using lots and lots of oxygen, at some point your peak or the set level of oxygen within your blood is going to decline, which is called oxygen debt. This is why breathing and heart rates rise as you work out so that you can get that oxygen back into your blood. The oxygen at the tissue sites is then able to convert lactic acid back into glucose and glycogen for your cells to use either in aerobic or anaerobic metabolism. This can also help to remake creatine phosphate and replace oxygen that has been lost from myoglobin. Myoglobin, as I mentioned, is that oxygen-storing pigment molecule in those muscle cells. Oxygen also helps to regenerate ATP. Oxygen replenishes ATP stores that were used to run other reactions in the body, sped up by increased body heat. Oxygen also speeds the heart and breathing muscles to use more ATP. The presence of oxygen is also going to replenish ATP that can be used in tissue repair processes. This animation reviews muscle metabolism. Muscle contractions aren't always on a massive level. Not always are all muscles contracting at one time. In other words, sometimes just single muscle fibers are contracting. And this is called a twitch, right? We've all sat there sometimes, even in class, when your eye muscles are twitching and it's so annoying, or maybe you get a twitch in your calf muscle, for example. A twitch is just a single muscle contraction, and that's in response to a single nerve impulse. There's 3 periods to this twitch muscle contraction: a latent period, which is kind of a building period just before the contraction happens. Then there's a contraction period, and then there's a relaxation period. Notice the time in milliseconds. It takes almost 30 milliseconds, less than half a second, for these contractions to occur. So they're very, very rapid, which again you would be aware of if you've ever had these or remember having these types of twitch reactions. Tetanus is a longer sustained contraction. This is going to develop more force than a single twitch. There's 2 ways to get an increase in force. One is called temporal or wave summation. This is when there's an increased number of frequency of impulses. So one action potential comes followed by another and another and another and another, and that's just going to cause the tension to build because the signal keeps being amplified from the nerve cell. Another would be called motor unit recruitment. This is going to increase the number of motor units or neurons that are stimulated at a single time. So instead of just sending the information down one neuron, the brain is going to send the information down several neurons to get a more sustained or higher rate of contraction. So twitch can eventually reach tetanus. Here you can see a single twitch, and you can imagine this graph continuing, just a quick one signal from one single neuron. Wave summation would be that action potential keeps coming and coming. So here's 2 action potentials rapidly behind each other. And then in an unfused tetanus or incomplete tetanus, that tension is building as the action potentials are coming down from the nervous system. And then finally a fused tetanus, which is where the muscles are not allowed to relax at all. This would be in the case of, say, lockjaw. Whereas an unfused tetanus would probably be like a Charlie horse or a very bad muscle cramp. This animation here goes through the control of muscle tension. There are 2 different types of muscle contractions. Isotonic would be where the muscle length changes, so lifting or lowering your leg, or bending your arm to flex your biceps. That would be an isotonic contraction. An isometric contraction would result in tension that's generated, but there's no movement. The muscles don't change in length. So you can sit there, I'm assuming you're sitting listening to this lecture, and you can tense up your quadriceps muscles or your biceps muscles without actually moving your arm or moving your leg. That's an example of an isometric contraction. Or your arms pushing against an immovable wall. That would be another example of an isometric contraction. Muscle tone is the general term used to describe how contracted at a resting state your muscles are, or how ready to contract they are. We use this idea of tone pretty much in the same way in our everyday language. Some people might say, oh, I don't really want to lose weight, but I just want to tone up, right? It's the same idea. You want your muscles to be more defined. You want your muscles to be tighter. The less you use your muscles, the less tone they're going to have, the more flaccid they're going to be at rest. So a small number of motor units are involuntarily activated to produce a sustained contraction. You may have heard that the more toned your muscles are, or the more muscle mass you have, the more you're going to burn calories, or the higher your metabolism. And that's true, because if you just look at that statement I just read to you, those muscles are being involuntarily activated or contracted even at rest. So when you have toned muscles, your muscles are maintaining some type of contraction or tension even when you're resting. So your metabolism is automatically going to go up to keep those muscles contracted. This can occur in skeletal as well as smooth muscle. So even the muscles of your intestines and stomach, for example, can have a certain level of tone to them. Smooth muscles in the arteries and veins also have some muscle tone to them, so they can maintain a certain level of blood pressure. Maintenance of normal posture by skeletal muscles is also going to involve muscle tone. So it's not just those toned arms or toned abs, but it's also toned back muscles to keep your posture upright. There are two main types, or really three main types, of skeletal muscle fibers. So we talked about the different ways they can contract, but all skeletal muscles aren't the same. Some have small diameters and some have large diameters. Those that have small diameters and many mitochondria and appear very red because of lots of myoglobin are called slow fibers, or SOF, slow oxidative fibers. ATP is generated mostly through aerobic respiration, and that's because of all of that myoglobin and all of those mitochondria. That myoglobin, remember, stores oxygen, so it's going to make lots of oxygen available to the mitochondria for ATP and aerobic respiration to occur. These muscles are going to develop tension very, very slowly. They're very resistant to fatigue because there's lots and lots of mitochondria and lots of oxygen stored in them. These muscles are capable of prolonged sustained contractions. These would be the types of muscles that are associated with our posture, our back muscles, our abs, that are constantly in a state of flexing and extending because they need to be that way for us to stand upright. Also, individuals that are training for marathons or doing kind of endurance type events are training their slow oxidative fibers in the process. There are also fast oxidative glycolytic fibers. These are kind of intermediate diameter. They have large amounts of myoglobin. They do appear red. ATP is generated mostly through aerobic respiration as well as glycolysis, which remember is anaerobic. They're moderately resistant to fatigue, and these are associated with larger motor skills. Finally, there's fast glycolytic fibers. These are large and white as opposed to red. Their white color means that they don't have much myoglobin, if any at all, which means they also don't have lots of oxygen. Without oxygen, you can pretty much bet that the majority of their metabolism is going to be based in anaerobic respiration. They have large amounts of glycogen. The ATP is generated mostly through glycolysis, which remember is anaerobic, and because of all that anaerobic respiration, they're going to fatigue very quickly. This also means they can contract and relax very quickly, so they have a short surge of power. These would be sprinting muscles. These would be muscles involved with fine motor skills. This is why after a very long lecture or a long essay, your hand is cramped up because those muscles of your hand, which are used to fine specific motor skills, aren't used to writing for, say, an hour or so. Skeletal muscle fibers in general, the majority of them are slow oxidative fibers. The rest of the composition varies depending on the muscle. Arms and shoulders are mostly fast glycolytic muscle fibers. The muscles in our legs are more slow oxidative or fast oxidative glycolytic, depending. Individuals' genetics will also determine the types of muscle fibers that make up their muscles. And this, some people are naturally faster than others, whereas some people have a natural gift for endurance running more so than others. It doesn't mean you can't train to be fast or train to be a distance runner. It just means some people have a natural aptitude simply because of the types of fibers in their body. Motor units contain all of the same fiber types, so they're recruited in specific orders depending on the requirement of speed, force, and duration of contraction. Let's stop here for